with the process known as meiosis. Meiosis is not mitosis. You don't want to get those confused because they're two completely different things. Meiosis occurs in stomatic or sex cells, while mitosis is body cell. Mitosis has one phase of interphase followed by one stage of cell division, which is prophase, metaphase, anaphase, and telophase, followed by cytokinesis. Okay? Well, meiosis has one stage of interphase, like mitosis, followed by, in addition, two phases of cell division. Prophase, metaphase 1, metaphase, anaphase, telophase 1, and then prophase, metaphase, anaphase, and telophase 2. But the names are important, so you don't have to worry. So then we start with meiosis. So it grows, develops. You have these homologous pairs that are identical. Remember, these two are two chromosomes. They are similar, but they're not exactly alike. That means the alleles are not exactly arranged they're not identical, but they are similar. They code for the same similar genes. So then you go through G1, it grows, develops, okay, these homologous pairs. So then you go through S phase. S phase is when they replicate. So then when they replicate, these become single duplicated, double duplicated chromosomes, forming these chromosome pairs, homologous pairs. Alright, so let me put these for a second. So then as it develops, you have G2, which is finishing development, and then you start the first cell division. After interphase is prophase. Now prophase, the first step is that this nuclear envelope disintegrates. It's gone. So then, the spindle fibers form, the centrioles, and then they start to move, these centrioles start to move to the other side of the cell. On the other side of the cell. So then, one of the most important things that occurs in prophase 1. This leads to a significant increase in genetic variation, and this is not found in mitosis. So you gotta remember that. The first step, this is known as crossing over. So then these homologous chromosomes come together to form this tetrad, or a group of 4. This tetrad, at the chiasmata of the tetrad, the alleles, or the DNA, is exchanged between the mother and the father. Alright? So it's exchanged between the mother and the father. So since this is exchanged, these are switched. Alright, this is a good. So then this increases variation because it's not found in mitosis, and this leads to the creation of four haploid cells that are genetically unique, which is not mitosis, because mitosis creates two diploid identical cells. All right, so let me finish switching these. All right, so let me switch these. All right, so now they exchange DNA. They are more varied, and so then to sum it up quickly, the nuclear envelope disappears. The centrioles move. Spindle fibers form. The centrioles are starting to move the other side, and crossing over occurs, which increases genetic variation. So then the spindle fibers, they already move to the other side of the cell. They attach to the chromosomes, and then they pull these poles. They pull the chromosomes into place. And that's metaphase plate. It's a metaphase plate where in the center of the chromosome, and then another form of genetic variation occurs here. This is known as independent assortment. Independent assortment means that these chromosomes are independent of each other. So the paternal and the maternal chromosomes, that came from your mom and your dad, are not dependent on each other. They're not like bonded or anything like of that sort. So then these could be arranged in different ways. These could be arranged like this. These could be arranged like this, and they could be arranged in many different ways. It all depends. So how did we get that? There's actually 8 possible ways the gametes could be arranged by independent assortment. The formula for this model, we have n equals 3, which means there are 3 total homologous pairs. So then you put in 3 for the n, so you get 2 to the third, which is 8 possible ways the gametes could be arranged by independent assortment. Second phase, so finally we get to anaphase 1, the first step of anaphase. Again, the names aren't important, just need to know what's going on. So in anaphase, or in this process, or in this step, finally the spindle fibers pull the homologous pairs to it. So these homologous pairs go to separate pools. Thus, anaphase 1, this is another difference, because mitosis, the chromosome, the duplicated chromosome is separated to each individual single chromosome. But in this first step of the split of meiosis, the homologous pairs are separated, not the chromosome itself, not the duplicated chromosome itself. So that's another difference between meiosis and mitosis. So finally, as the centrosome pushes in the chromosomes, or pulls in the chromosomes, that's the cytoplasm, it pinches in, and then finally it pinches in so much that this one cell becomes 2 independent haploid cells. Now, these chromosomes are already duplicated, so there's no need for another DNA replication, and the nuclear envelope usually does not. It kind of forms. Sometimes it does form, sometimes it doesn't. There's no exact way of determining it. It's all.
All right. We're now in meiosis 2, which is basically in this process, in this second stage of meiosis. Basically, the sister chromatids split from the duplicated chromosomes, and this is exactly the same thing as mitosis. And also, in this phase, which is also the prophase 2, the centrosome duplicates and makes another one, and basically the entire thing is starting over again. Centrosomes are splitting apart, creating spindle fibers. Sorry for the delay. Okay, and basically they're moving towards each opposite ends of the cell. And this is also happening at the same time to the other independent haploid cell. This also creates a second centriole, and then, it's not centriole, I'm sorry, centrosome. And then these centrosomes split apart, creating spindle fibers. And these will also go towards the other end of the cell. And okay, in metaphase 2, basically what happens is spindle fibers attached to the kinetochore of each in each chromosome, and this centrosome right here will move to the other side, and both the centrosomes will help pull the chromosomes into the middle of the cell, which is called the cell metaphase plate, and which is similar to mitosis. Okay, anaphase 2. Basically, what's happening in here is the each centrosome is pulling apart the duplicated the sister chromatids apart, and it's pulling towards, and the chromosomes are being pulled towards the centrosome. And as it is being pulled, it's forming, the cytoplasm is being pinched, and it's forming a cleavage furrow, and it gets pinched too much. Basically what happens is it gets pinched off, and they each become their own independent cells, and at the end you'll have 4 independent haploid cells. And I also got to mention that each independent haploid cell has genetic variation, so not one of these cells are alike. So
what if something goes wrong in meiosis? I mean, is this a perfect process? Of course not. One example, I mean, right now we're in metaphase 1 of meiosis, right before we're about to go to anaphase 1. So one example is called non-disjunction. In a non-disjunction, the homologous pairs aren't separated properly. So after anaphase, you'll have something very odd like this. As you can see above, the top cell, the future top cell, or the top part, is missing chromosomes, and this one has an extra male chromosome. So that's
one example of a non-dysfunction is Klinefelter syndrome. Klinefelter syndrome is when the person has an extra X chromosome, making what's supposed to be a man to be a more woman-like person. Some symptoms of this disorder include enlarged breasts, sparse body hair, small testicles, and such. Some treatments for this disease is daily injections of testosterone during androgen therapy, which is a therapy used to help viralize the person and help stabilize him. Also, another treatment for the person is speech therapy to help him with his slurred language, which is one of the side effects of Klinefelter's. And in addition, behavioral therapy to help him interact with society much more efficiently and help him live a better life. That's all, folks. That's the end.